can use it hopefully to, look at how we breed healthier dogs and how we can also use it to breed for certain characteristics, whether it's maybe coat, teeth, size, or whatever. I did send Dr. Dockweiler a set of questions as a guide, but also let her know that if there are things that Embark's doing that would be of interest or she believes would be of interest for breeders, please share that. So otherwise, without any other delay, I'm just going to hand it over to Dr. Dockweiler and let you get started. And then what I'll have you do is probably just jump in, and then maybe at the end if we can save some room for questions, because I don't want to, sometimes questions can derail a whole bunch of things in a flow. So if we could try to save most of our questions more towards the end, that would be awesome. So, anyways, Dr. Dockweiler from Embark.
Daryl, can I say something real quick, Daryl?
Yep, go ahead, Tim.
There's a raise hand icon at the bottom. If you have a question, if you'll raise your hand, and we'll try to answer as many questions as we can during the process. But we'll let the doctor do her speech and go from there. And then I just want to tell you that, Daryl. So back to you. It's your meeting. So
awesome.
Perfect.
Thank you. I'm so excited to be chatting with you guys today. I actually don't have any slides prepared for today. I wanted this to be a little bit more of a discussion. So a little bit of background about me. I've been with Embark for about 3.5 years now. Prior to that time, I was in private practice in theriogenology, which is a big fancy word for reproduction. So I do have sporting dogs myself, actually. I have Welsh Springer Spaniels, not quite as serious of hunting dogs as y'all's dogs are, but kind of involved in the fancy as well. So I'm first curious how you guys are using genetic testing currently. Like, what are you most looking at? What's the most interesting things to you?
I'll answer a little bit for me and then others can chime in. I'm not a breeder, but I do breed my dog quite a bit, and I'm always curious about a number of things. How can we improve coat? And I know that it has some information on coat, but it being a NAVTA judge, one of the things we see, or I'm starting to see a lot of dogs with poor coats, what we'd call an open coat, where they don't have good coverage. I'm not sure if you guys have a test that actually really talks about the density of coat, particularly coverage, but also looking at what health issues might be out there that might pertain to the breed. What are some of the things we have to be cautious about when maybe dogs have recessive genes and you consider, breeding them? I think COI is a really big thing for our breed because being a smaller breed and, years ago, Embark didn't have them yet in there. They probably didn't have enough database. So kind of what is a coefficient of inbreeding that might be too close or too high? And maybe why is there such a difference between like a 10 generation pedigree versus Embark, which obviously is genes, but a little bit into there. I know Kelvin had some things on like, does it have a test, or are you looking at tests possibly for bites, which may be a little harder when we look at overshot, undershot, butt bite, and cross bite or parrot mouth or whatever they would call that. And then whatever other people may want to add to that.
Well, awesome. Well, my first question on my list here is what genetics can and can't tell us about our dogs. So you put on a few of each of those actually with that that little summary there. So genetics can tell us the risk for known disease-associated mutations. So the known part of that is really important. So if we haven't discovered what underlying genetics are going to predispose to a disease, then it's not something that we're going to be able to test for. And then kind of the flip side of that coin, if we do have a known disease that we're testing for, say progressive retinal atrophy. So that's a type of blindness that can occur over time. There's a variety of different genetic mutations that cause that disease, but all of them are going to lead to blindness. So just because a dog tests clear for the known progressive retinal atrophy variants doesn't necessarily mean that they'll never go on to develop that disease, because there are always known unknown mutations that could potentially lead to the same or a similar disease process. Oh, sorry, I have a question. Oh, PRA stands for progressive retinal atrophy. So that's just an eye disease. It's a non-painful loss of the vision cells that happens over time. So there are a variety of different types. Some of them occur at very young ages. Some of them occur at very old ages. But yes, thank you. I will define my acronyms moving forward. I'm sorry. So even if a dog does test clear, it doesn't necessarily mean that they are 100% guaranteed never going to get a similar disease process, because there always might be a new genetic mutation that pops up within a population that's not going to be something that we can test for. And then the other thing that genetics may or may not be able to tell us about are complex traits. So that would be things that have either multiple genetic mutations that underlie that disease process, or perhaps interactions of the genes with the environment. So a really great example of such a disease would be hip dysplasia. So I'm sure everybody is probably pretty familiar with hip dysplasia. But the underlying cause of hip dysplasia, or abnormal development of the hip joints, is actually joint laxity. So if you think of the ball and the socket, if that ball is not fitted very tightly into that socket and it moves around inside that socket, it'll cause arthritis. So that's usually what we're looking for on like an OFA X-ray. So there are probably a lot of genes that contribute to that hip laxity, or that looseness of that hip joint. But we also know that there are some environmental factors as well. So nutrition when puppies are really young, things like doing stairs with very young puppies or raising them on very slick floors. That's actually the reason we don't like to use newspapers when we're raising puppies anymore. Even though it's very easy to clean, unfortunately, that slick surface as they're learning to walk is not ideal for their hip joints. So that's not going to be something that's easily genetically testable at this time. Now, there are in the dog becoming more what's called polygenic risk scores, which takes a bunch of different genetic locations and kind of takes all that information together and provides a risk that your dog may go on to develop that disease. But those are really in their infancy at the moment. So we have a few that are kind of at our disposal, but they're really not in a good place to make breeding decisions on at this point. And they're still being developed into the future. All right, how does the Embark testing control process work? What's the percentage of contaminations in tests that can lead to inaccurate results? That's a great question. So the first part of that question that I'll answer is the contamination. So if we do have a swab that has DNA from multiple dogs on it, that's actually something that's really, really easy to catch in our quality control process, because we'll have more than 2 complete sets of genotypes. And actually the test just won't run. It'll come back as an error, and we'll have to re-swab the dog in order to get results. So contamination is not, like, if you get results, the swab has not been contaminated, because it would fail earlier in the process before you actually receive those results. As far as quality control, there's actually no oversight of veterinary genetic testing labs in North America. So it's really up to the individual company to make sure that they're holding themselves to a very high standard. So at Embark, we are CLIA approved and ISO certified. So that means that we follow the same strict quality control guidelines as a human lab would and is required to be upheld to. And actually, our lab used to run some of the human genetic samples as well. They're not currently, we're their biggest client now, but they did used to run some of those human tests, so they are a human approved laboratory. All of our swabs are barcoded. It's not a manual process at all. It's all a machine that reads it and handles it. So it's very, very well controlled, but that's a great question. All right, any other questions before I move on to the next in my list? All right, and you had asked Daryl about COI as well, and kind of how to apply. How can a Pudelpointer come back with 5% GSP when we know there is no GSP? That's also a great question. So if we do have a small amount of another breed that shows up on our testing results, that actually could potentially be a positive thing. So the way that we assign breed ancestry on this genetic test is we're looking at very long stretches of DNA that are identical between individuals of a single breed. So as those get shorter and shorter, we can assign those percentages to an individual dog, and that's how we end up with the percentages, especially like with a mixed breed dog. So we do have a pretty good reference panel of poodle pointers in our database, but of course it's not perfect. So if we do have a small amount of other breed that comes back, that could be representative of genetic diversity that's not actually captured in our panel, which potentially could be a good thing. And that actually leads really nicely into my next topic here, which was going to be coefficient of inbreeding, or COI. So the coefficient of inbreeding just is the level of inbreeding of an individual. So that's usually going to be measured or represented as a percentage or potentially a decimal. We represent it as a percentage on the Embark test. So there's a couple ways that you can calculate that. You can either use a pedigree-based COI, or you can use our genomic-based measures. So like Daryl mentioned, the pedigree-based and the genomic-based can vary really, really widely. So pedigree-based, you're going back 5, 10, maybe more generations, depending on the breed club and how on top of it you guys are with record keeping. Of course, that does depend really heavily on record keeping accuracy and nobody fudging any numbers in the past, if you will. That's going to assume that 50% of the DNA came from each parent, 25% from each grandparent, 12.5% from each great-grandparent, and so on and so forth on back. That's actually not quite true in biology. That's because of something called recombination. So that's where the genes shuffle around a little bit. So each individual is going to inherit 50% of their genome from their mom and 50% from their dad, but it may not necessarily be exactly representative of 25% of each grandparent. And I think a lot of breeders do actually recognize this, as one grandparent is sometimes more either phenotypically or behaviorally or whatever expressed in an offspring than you might expect. So that's why that's genetic recombination. The other thing with those pedigree-based COIs versus the genomic-based COIs is that the genomic-based are going to go all the way back to the beginning of time. So when that breed was founded, and oftentimes founding members of a breed were related to one another. And I think you guys were founded from like 9 poodles and 50 pointers or something like that, right? So kind of a finite block of dogs, if you will, and potentially some of those pointers and some of those poodles may have been related to one another. So you can go back to the beginning of the breed all the way with a pedigree-based COI. That's just not something that's ever going to be taken into account. So the genomic-based COI is a little bit more accurate in terms of true levels of inbreeding. But it is important to look at your pedigree-based COI as well, because there is some evidence that more recent inbreeding can be a little bit more detrimental compared to that historic inbreeding. And the reason for that is because we've had a lot more time with that historic inbreeding, the fact that those 50 pointers may have been a little bit related to one another. We've had a lot more time for some of those things to kind of reveal themselves, to sort of shake themselves out, versus if you just bred a brother and sister together, today. So much more quick bringing together of potentially some recessive genes in that circumstance. So the important thing to know about COI is that we do need some to make those consistent, desirable characteristics of our breed. So it's not all bad. So then people always ask me, okay, well then why do I care about this COI thing if we need some of this? And we care because of something called inbreeding depression. So that's the reduced fitness of offspring of very closely related individuals. So in the dog, we've found that leads to decreased litter sizes, increased neonatal death, increased disease prevalence, and decreased lifespan. So all bad things, of course. There was one paper that found for every 10% increase in that coefficient of inbreeding, we had 1 fewer puppy per litter. So obviously not super ideal. So question is, what COI percent is considered inbreeding versus line breeding? And the population geneticist answer to that is there's no real distinction. So line breeding is a nicer way, I think, to say inbreeding, and it is a way to concentrate some of those desirable traits that you want, that you're looking for, maybe hunting ability for you guys, thick coat, things like that. But it's not necessarily distinct from inbreeding, if you will. So the other important thing to know about inbreeding is that the COI of the offspring depends on how related the parents are to each other, not how inbred they each are. So if you have two very inbred individuals, if they are distantly related, they potentially could produce puppies with low coefficient of inbreeding. And then the opposite is also true. So if you have two very outbred individuals and their brother and sister, you breed them together, you're going to result in a litter with a pretty high COI. You also maybe noticed, do you guys do any puppy testing of your pups? Yeah. So you've probably noticed as well that the COI is going to vary a little bit based on the puppy. And that's because each puppy does inherit 50% of their DNA from mom and 50% of their DNA from dad, but it's not necessarily the same 50%. So some of them will have a little higher inbreeding percentage. Some of them will have a little lower inbreeding percentage. And there are some breed groups that use that to try to increase the level of inbreeding within their population. So the next question here is, what do you consider a high COI? And it really depends on what you're seeing. So there's no like one number to tell you to shoot for. Historically, people do try to stay under about 5% with 10 generation pedigrees, but that's going to depend on how closely related the breed is, what type of population we have to work with, that sort of thing. So I actually looked up your guys' numbers, and your average COI for Embark tested poodle pointers, which again is just the Embark tested ones, that may not be totally representative of the entire US or world population of this breed, but is at 17%, which is actually quite good. So there are breeds that are much higher than that. My breed's at about 28%. It's a pretty small breed. You guys are also a pretty small breed. So the fact that you've maintained at 17% is really great. The most inbred breed is the Norwegian Lundehund. They're at about 89% coefficient of inbreeding. So they're doing some things to genetically rescue that population, because that, of course, is not sustainable.
Wow. So if ours is 17% and breeders are trying to make decisions, if we say 17% is the baseline, are you saying that 5% over 17% would be kind of a norm to look at that you wouldn't want to go over? Because when you said 5% is kind of that key.
5% based on pedigree. So the genomic is always going to be a little bit than that. Yeah. So we try to keep it under 5% when we're looking at pedigree, because that is the more recent inbreeding, which potentially could have a higher effect than that historic inbreeding. But generally, once a stud book is closed, which I believe is your guys' situation, correct? You're not bringing in other pointers or other poodles. It's just breeding to within the same population. Is that right?
In the US, yes, but in Germany and Europe, they do nick the lines with pointers still to this time, I believe.
Okay, so then there potentially could be potential to bring in new genetics, depending on their registration requirements and all of that. But generally, once that stud book is closed, the best that we can hope for is really to maintain that level of genetic diversity that we have. So doing pairings, we can use Embark to do the pair predictor, just automated software to get estimated COIs. Of course, that is an estimate. Like I said, they're going to be a little bit different on either end, some a little higher, some a little lower, but trying to maintain within that 17% is a pretty good benchmark. And do you guys see anything like, high cancer rates or high rates of specific diseases that maybe we can't test for, or like small litter sizes or anything like that? Or are we doing pretty well from a population genetics standpoint?
I don't know if Alicia is on to answer that, because I know she's been collecting some data on that.
Yeah, I just got on. Sorry for being late. Yeah, there's nothing, I would say nothing significant at this time, but we are starting to track, incidences of cancer. Small litters hasn't really been, I don't know that it's been a big thing for many people. I know some people are seeing small litters.
Yeah.
But yeah, we're just now starting, or just in the last year or two, starting to try to really kind of collect that data. We've got a member survey, or it's really not just members, it's open to anybody, to try to, see how much cancer we're seeing and stuff like that. So we are starting to track that, so that hopefully in the future, we can, partner with you guys and explore that if we need to.
Yeah. Yeah, for sure. That would be awesome. And the surveys are currently paused within the Embark platform, the Embark Volunteers platform specifically. But when they're back online, I encourage everybody to fill those out. We actually use surveys to then connect diagnoses to genetic variants to try to come up with new tests and things. So that'll be really important once we get those surveys back online. Yes, so the next question here is there forecasting available that allows a breeder to underpin what COIs will be produced by potential pairings? So yes, and we can do that via a pedigree analysis. Most pedigree softwares will automatically calculate that for you, and you can choose the number of generations. And Embark does calculate the genomic estimated COI as well. So we do have that for you. It's under the Pair Predictor software. Oh, we see some urinary issues in puppies. What urinary issues in pups are we seeing?
I would say generally UTIs in the females a lot, and we do have, I mean, I have seen incidences of the hooded vulva. But yeah, for some reason, female poodle pointers as puppies, we tend to get a lot of UTIs.
Interesting. That's strange. That's probably more a consequence of being a female puppy and potentially having long hair in that area than necessarily related directly to the poodle pointer. But that's interesting. I'd be curious to see how that shakes out for sure.
Yeah, if anybody else on the call has had other urinary infection or issues, please, feel free to share.
And I've been told, I don't have a particularly long-haired breed, but other folks have told me trimming the hair on the vulva, like when they're little puppies, can help with that kind of thing. So that might be something to try as well.
I think another factor there is we tend to introduce dogs to water very early.
Yep.
And,
also
they're playing in the mud and swamp and that sort of thing. So
yeah, that's probably more of a dog thing than specifically related to this breed in that case. But that's good. That's good. No UTIs yet. Excellent. I love to hear it. All right. I think I answered this one. All right. So I do have a question on my list here about interpreting a carrier result for autosomal recessive conditions when we're considering a mating. So, and the follow-up question is it ever acceptable to breed two carriers? And really the answer to almost everything is going to be, it depends. So recessive diseases require two copies in order to be at risk for that disease. So there are a bunch of diseases that fall into that category. I would say most of the genetic diseases that we're able to test for do fall into the recessive category. So that means that a puppy has to inherit one copy from mom and one copy from dad in order to be at risk. So using carriers within your population is totally appropriate and a great thing to do as long as they're, otherwise breeding candidates. And I know you have strict rules on abilities and all of these things to make sure that they're otherwise good breeding candidates. Totally appropriate to use them. We're going to want to breed them to a clear individual when we can do so. So about half of that litter would also be carriers of that condition, but we're not going to produce any that are at risk. And people sometimes ask me, well, we just want to get this disease completely out of the lines. But the problem with doing that is then potentially you're narrowing that gene pool more. You might introduce something that you can't test for, which is, of course, a much less controllable set of circumstances. Breeding two carriers together, definitely not advised if we can avoid that. However, breeding an individual that is at risk themselves could potentially be appropriate. So if you have a dog who's at risk, let's say for one of those progressive retinal atrophies, so causes blindness, provided that breeding isn't going to exacerbate their condition, they can also be bred to a clear individual. So that litter would be 100% carriers. So all of them are going to carry that disease, but none of them would be at risk for that disease. So that could potentially be an appropriate tactic to use. Any questions on that? All right, awesome. Oh, and there's the question here about which portion of the test should we prioritize most when we're making breeding decisions? And again, I know this is very frustrating, but the answer is it depends. It depends on the individual. It depends on the goals of your breeding program. It depends on where you want to go as a breed group. So if you had a specific disease that you were really working hard on getting out of this breed, then potentially specific disease markers would be the answer to that question. If you really wanted to work with furnishings and make sure everybody has a good coat, a nice thick coat, then that potentially might be what you prioritize. If you're starting to see things like your COI is creeping up, if you're starting to see small litters, shortened lifespans, then COI might be the thing that you consider. It really depends on what you guys are seeing as a breed community.
So when we look at coats, because that has been a conversation we hear frequently, how can Embark help us look at coats and try to improve coats in the breed, or at least have more consistent coats?
So the coat markers that we test for are just single locus, so just one gene that affects like long hair, furnishings, color, that sort of thing. So probably a better tactic to get proper coats, which I know is important for you guys for protecting dogs from brush and things, would be to look at just do like a pedigree analysis rather than looking at those single genes, because there are probably other genes that we don't know of yet that control more things like density, skin thickness, like the strength of the hair shaft, like things that we're not able to test for yet. But if you could include like in your pedigree software, that might be helpful. If you have like you call it open coat versus nice dense coat, that might be something helpful to track so that you can breed hopefully towards that.
Okay.
I have a question on the process of doing a DNA analysis. So I assuming you're looking at just nuclear DNA, correct?
Correct.
And so how do you choose what segments, what portion of the genome you want to compare?
For like diseases or for COI or for all of the above?
Say for anything. I mean,
Okay.
well, not so much for a disease. I guess if you're looking for a disease, you would do that, but particularly say for COI.
Okay, yeah, awesome. So the way that the Embark test works is it's called a microarray. So that looks like a little microscope slide. It's got many hundreds of thousands of little beads on it. And on each of those beads, we have several probes. So those are short fragments of DNA that would match to the dog's DNA sample. So when you swab your dog's cheek, you're collecting a bunch of cheek cells, which is where we get that nuclear DNA from. So from there, the cheek cells go to our lab. That DNA gets amplified, or many copies of it are made. It gets unzipped, that double helix, we rip it apart and chopped up into little pieces. And then that patient sample is bathed over that little microchip, and then complementary base pairing occurs. So the DNA binds to those little fragments. So as far as where those probes are located, we do have some of them pointed at diseases, which like you say, I mean, those are published. We know exactly what DNA sequence leads to which disease. Same thing with the coat traits. So we know the gene that causes furnishings. We know the genes that cause long hair. So we're going to have probes pointed at that. Other things are going to be pointed at ancestry and other areas of interest in the genome to make discoveries. So when we're looking at COI, we're looking at long runs of sameness between two dogs, if that makes sense. So we have over all of the chromosomes, over all the X and Y chromosomes as well. So we can actually make genetic sex calls and determine, in some cases, disorders of sexual development as well. But many hundreds of thousands of probes are a lot of different locations in the genome. I hope that.
Are you applying AI to any of this? And also, along with that question, in terms of determining epigenetics?
Yeah, great question. So AI is not applied currently to our pipeline. It is an automated pipeline, but it's not artificially intelligent in any way. Like a human designed the pipeline in order to spit out the ancestry results and the trait results and the health results as we've designed it. And we do actually, we're dipping our toes into methylation. So we do have an age test. It's based calendar age test. So DNA methylation, if you think about you have DNA that's inherited from your parents, that doesn't change throughout your whole life. That's going to be the same on the day the dog is born till the day the dog dies. No changing throughout life. But the way that that DNA is expressed or how it's read by the animal can change throughout life. And that's what DNA methylation is. So you can think of it as little tags that are on the outside of the DNA that turn on and turn off certain genes. So for age, that methylation does occur in a predictable pattern. So some sites will increase in methylation, some sites will decrease in methylation, depending on age. So we do have an age test available. It's about as accurate as any other calendar age test that's on the market currently. So we can predict true birth date within 12 months for about 77% of dogs. But that technology has a lot of potential for both disease, for aging, for healthy aging, but it's really still kind of in its infancy, especially in the dog. All right, can any of the traits tested for be sex-linked? So the traits currently that we're testing for, when I say traits, I mean like coat color, furnishings, that sort of thing. Those are not going to be sex-linked. So those are all going to be autosomal. So we have the autosomes and then the sex chromosomes. Those are like the two types of chromosomes. But there are some diseases that are certainly sex-linked. And usually those are going to be X-linked recessive. So females have two X chromosomes, males have an X and a Y chromosome. So essentially what that looks like is females are typically asymptomatic carriers of those conditions, but males only need one copy to be at risk because they've only got one X chromosome. So those diseases tend to be quite nasty. So we don't recommend breeding carrier females because half their male offspring would potentially be at risk for the disease. Those are normally like hemophilias, really bad skin diseases. They tend to be really nasty and life-limiting, but they also fortunately tend to be rare. But that's a great question. All right, is there a gene test for heart conditions, particularly PDA, murmurs, aortic stenosis, et cetera? So we have done some work on both dilated cardiomyopathy and tricuspid valve dysplasia. I know those aren't diseases that you guys have asked about here. But currently there's no simple genetic test for any cardiac condition. But if you do have puppies that have been diagnosed with those conditions and have been Embark tested, or even if the parents have been Embark tested, filling out those surveys and letting us know when we have those diagnoses can hopefully help us make some genetic links. I would guess that most of those are going to be more complex than just a single gene leading to the development of the disease. It feels like we would have found those already if they were findable in that way. So it probably ultimately is going to look more like a polygenic risk score or a more complex prediction. All right, what traits are actually testable or predictable? That's not a novice question. That's a great question. So we can generally predict size, not probably for you guys as a purebred breed. It's not like a huge variation in size anyway, but we do have a genetic weight algorithm that can predict size. About 80% of size in dogs is controlled by only about 7 genes. Compare that to like 20% of height in humans controlled by 700 genes. So it's a lot simpler in dogs than it is in people. So we do have a genetic weight algorithm. We can test for various coat traits like coat length, coat color, most coat colors. Some are not completely ironed out yet. We can test for certain types of white spotting. Not all types of white spotting, unfortunately. And we can test for furnishings, which I know you guys have, comes from the poodles, which is kind of that wiry coat texture that you guys have. Other things that we can test for are certain types of dwarfism, which are common in certain breeds. So that's like a short leg, long body appearance, certain types of blue eyes, certain types of hairlessness, double dew claws. There are a variety. We have about 50 that we can test for currently. Ooh, copper toxicosis. Yes. My favorite and least favorite disease of all time. So yes, I would love to talk about copper toxicosis. So in May of 2024, we started testing for 3 copper toxicosis variants. Those are ATP7B, which is the accumulating or bad variant, ATP7A, which is one of the attenuating variants, and RETN, which is the other attenuating variant. So both of those attenuating variants can partially offset the bad effects of that ATP or accumulating variant, ATP7B accumulating variant. So copper toxicosis is a pretty complex disease. Any dog can develop it regardless of genetic predisposition or not. It depends on how much copper they're taking in their diet and also in their water sources. So there is some well water that's very high in copper. There also are some older homes still that have copper pipes that some dogs will get an excessive amount of copper simply by drinking the water that's coming from those copper pipes. Fortunately, less common these days, but still does sometimes occur. So any dog can develop it. Some dogs we know are genetically predisposed to developing it. So that ATP7B variant that I mentioned, the accumulating or the bad variant, we know that that increases the risk of disease in the Labrador and the Doberman Pinscher. The jury is kind of out on all of the other breeds that are out there. So we're still researching that. Research is ongoing, but we haven't proven a direct causation between that variant and the development of copper toxicosis in any breeds besides the Lab and the Dobie. So what copper toxicosis looks like, it's pretty hard to diagnose because it does need a liver biopsy with a special copper quantification. So it's not even just like a regular liver biopsy. It has to be sent for special testing, special staining. And the symptoms are very non-specific. So oftentimes we'll see elevated liver enzymes first, like on a blood test. If you're going in for routine blood work, perhaps your liver enzymes are elevated. Maybe the liver will look a little bit funny on ultrasound. Maybe the dog will be a little bit off, maybe not want to eat, have some vomiting, diarrhea. In the very late stages of disease, we start to see things associated with liver failure. So that could be ascites or fluid in the belly, yellow mucous membranes or like yellow whites of the eyes or jaundice and swelling of that liver. So that's generally in the very late stages. So it's kind of hard to prove that you have copper toxicosis because a lot of us will try treatments before going in for a biopsy, which of course is pretty invasive, has a bleeding risk. So hard to prove, but there are certain treatments that can bind that copper and help excrete it with the feces rather than having it store up in the liver. So we don't know if these variants have an effect in this breed. We also wouldn't be in this situation in the first place if the dog food companies in this country had a cap on the amount of copper they're allowed to add to their foods. So essentially they have a minimum that they have to meet for copper because of course that is important for growth. It's important for maintenance. It is a mineral that's essential for life, but they don't actually have a maximum. So it's hard to determine how much copper is actually in a food. You generally have to call the company to see. It's not going to be like printed on the bag. And if they just had a maximum, then potentially even dogs that were genetic risk wouldn't even have problems with this. So for breeding recommendations for you guys, we don't recommend taking it strongly into account at this point until we know if it leads to disease in this breed. If you have a dog that has 1 or 2 copies of that ATP7B, it might be worth calling the food company to see how much copper is actually in that diet, to see if maybe we can get on something that's a little bit lower copper if they happen to be way above that minimum that AAFCO publishes. But yeah, that's a great question.
I have a question that you kind of alluded to there that we haven't talked about much, and that is breed specificity. A dog can have a genetic test for a disease, and in certain breeds it just does not occur.
Yep.
So it seems to me, in order to really make good sense out of genetic testing for disease, you have to get the phenotype information. So how do you do that?
So that's what we collect through our survey data. But that certainly happens, and that's been published. It's known degenerative myelopathy, which is the dog version of Lou Gehrig's disease or amyotrophic lateral sclerosis, is a really good example of that. So German Shepherds have like a 5% chance of testing at risk, but they're like the poster children for that disease. Like if they have 2 copies of that recessive variant, they're probably going to develop the disease. But wire-haired fox terriers, almost 90% of them are at risk for disease and almost none of them develop it. So there's some protective factor or there's some other unprotective, I guess, a facilitating factor in the German Shepherds that lead to that disease in those breeds. So yeah, we really rely on you guys to report the phenotype data to us through those surveys.
And is there, I know we have a small breed and probably small numbers being tested, but is there any way you can communicate back to us that breed specificity in poodle pointers?
Like for the copper toxicosis specifically?
No, not well, that's
just
just one
general?
example, but for various tests you have, for instance, progressive retinal atrophy.
Mhm.
I think that's very rare in a poodle pointer. I've never
Yeah.
heard of anybody having it. And yet there's a test for it. So, you could say, well, we don't really care about it until somebody starts getting it. So what I'm saying is if you give out the test, then pair it with some phenotypic information. How common is that disease in that breed? And that would have mean a lot more.
Yeah, oh yeah, for sure, for sure. And as we did make those connections and make those discoveries, we do move diseases around. So I'm sure you've seen on your Embark account, but we have breed relevant and other. So breed relevant means we know that this disease, this variant is very likely to cause disease in this individual or in this individual breed mix. Other means it's either not been proven or has proven not to cause disease in this breed or breed mix. So we do parse those out for you so that you have
Okay.
all the information, but not necessarily going to pay too much attention to the other category until we know more. But we do change them around as we discover new things. Absolutely. All right, I have a few other questions. Let's see. Once DNA has been submitted, who owns the genetic data, me or Embark? So you own your own genetic data. You can download your raw data at any time. There are some probes that are proprietary, so those won't be listed on the raw data, but you can certainly delete your account. You can delete the pet at any point. So that certainly belongs to you. All right, next question. I've seen substantial differences in coat seasonal loss, shedding, while some coats change mildly. Others suffer significant shedding with spring, summer conditions in both of their double coats. Can the propensity for shedding be identified? So we do actually have one locus that we're looking at for shedding currently, but we do have some other scores that potentially could be like a polygenic score for shedding. And there's a lot of other factors at play there rather than just genetics. So like area of the country in terms of how cold it is and how hot it gets. So how much we build up that coat simply because it's cold. Day length also has a lot to do with it. So folks who live further north have a bigger swing in that day length than people who live further south. So that can affect it. Hormones as well. So spay neuter status or seasonal cyclicity of the females especially can certainly affect shedding as well. So it probably won't ever be something that's perfect, but we're hoping to get closer on the shedding front because I know that would be really useful to a lot of people. All right, how many affected individuals would typically be required to be able to confidently link a marker to a disease or a trait? And the answer here again is it depends. So if we have a very closed, like single family, like we know we bred these two dogs together and 25% of this litter is affected, then we can pretty safely assume probably both of those parents are carriers. Breeding them together is what resulted in that 25%. So we might not need more than just that one family. It's generally helpful to have the affected individuals, those affected individuals' immediate family members, and then potentially some unrelated dogs so that we can make comparisons and see where things might be different. If we're talking about something that's less well characterized, maybe only pops up occasionally, usually we need hundreds of individuals in order to draw any kind of meaningful conclusions. And then add to that the complication of the environment plus the genetic susceptibility. Of course, that makes that even harder.
Politician.
All right, see if I have any on my list here.
Yeah, I was gonna say we're running through the questions pretty
Yeah,
good here. You
yeah,
guys are
no,
coming
this
up
is
with
great.
great questions. This is really cool. This is great stuff.
All right, let's see. There is one here about common misconceptions breeders have about using genetic testing in breeding decisions. And I would say the biggest misconception that I run into is because a dog is clear means they're absolutely clear. And that's like the only thing you need to be a good breeding candidate, which I know that your breed group doesn't ascribe to that philosophy, because I know you have working tests and you have other health tests and things that you require, which is great. But that's probably the biggest misconception that I run into. And like we talked about before, just because they're clear doesn't necessarily mean they're never going to develop the disease. Doesn't necessarily mean that we're not going to pass something down that we maybe aren't aware of that could potentially be harmful. And then there's a question here about breed clubs using aggregated data to make decisions at the population level. So I know you guys are a breed club partner, so you do get that breed club report. The other thing you could consider doing if you want to use all of your dogs together on like the pair predictor tool to do COIs and stuff, is if you're up for it, creating one club account and then sharing dogs to that account, so that everybody has like a shared login, everybody can get on that account, and everybody can use the pair predictor with everybody else's dogs. But of course, it's dependent on your level of comfort with
Who would we talk to about setting that up?
So you guys would just set it up like a normal account, and then we could help you. Yeah, we could help you share your dogs to it for sure. I would recommend setting it up with like a Gmail account so that you can use single sign-on. That'll be easy than trying to do the magic link thing with a bunch of different people.
Yeah. Okay. Well, yeah, we'll look into that for sure.
How can breeders best use the pair predictor part of Embark?
Probably for you guys that have a pretty good handle on diseases and using phenotypic data and things and using hunting ability, probably the estimated COI is going to be most useful for you, which it'll spit out all the coat traits, all like the expected colors and expected diseases for the things that we can test for as well. But I would probably recommend you guys use it as we have these two who are good breeding candidates, or maybe we have one female and there's 3 potential males, like who might be the best of these 3 males to pair her with to maintain that diversity. That would probably be most helpful for you guys. And then we have a question here. The Pudelpointer is not a part of Matchmaker. When will we be able to use Matchmaker versus Pair Predictor? I would love for everybody to be able to use Matchmaker. I'm definitely pushing that, but there do have to be a certain threshold of number of dogs tested in order to be eligible for Matchmaker, which I don't think the Pudelpointer has quite reached yet, but hopefully in the future, as more dogs are tested, we'll be able to have more available in Matchmaker. So that's where you can look at
on the website.
Oops, sorry, go ahead.
Playing with it.
I think that was just a
background noise.
Just noise. Approximately how many dogs do we need to get to that Matchmaker level that we need? I guess
I want to say it's around 500.
Okay.
But let me email you. For sure.
I think we're somewhere in because the first report we got was like, I think we were greater than 100, and then that was like 2 years ago, and then this year's report is like, well, you're greater than, I think it was like greater than 100 but less than 500, and I'm like, that's a pretty big spread.
I know, and I'm sorry, it's frustrating that we have to put the report out like that, but we don't have exact numbers because that potentially could lead to somebody getting information about a specific dog. So we need to be a little bit careful. But I did look at your population and it's hundreds.
Okay, good.
Designation these days.
We're getting there.
We're getting there, yeah.
I have another question.
Yeah.
Are you doing any work looking at maternal lineage via mitochondrial DNA?
Not specifically. So we do have some mitochondrial DNA information, but it's more from an ancestry perspective. Like this mitochondrial DNA line came from this area of the world rather than tied to specific diseases. There are not many specific diseases in dogs that are mitochondrial in origin or in inheritance. They do exist, but that's not something that we're specifically looking at this time. And then I do have a question here about what's the difference between Matchmaker and Pair Predictor. So Pair Predictor, you can use in any 2 dogs, but they both have to be on your account. So you can even do like mixed breed litters if you wanted to, or like that example of somebody's poodle pointer coming up with 5% GSP, that dog would still be eligible for pair predictor. You can use it with anybody. They just have to be on the same account. Whereas Matchmaker is you're looking at dogs owned by everybody who is opted into Matchmaker. So you can search like and sort by distance and things like that. It's a nice tool. But Pair Predictor is probably a little bit more flexible. And for a small breed community like you guys, probably works just as well. Ooh, is the Embark testing ever wrong? So nothing is ever 100%, of course. So what I can tell you is that our disease and trait mutation testing, those accuracy levels are well above 99%. So when we talk about genetic testing, we talk about accuracy. And what that means is, did we detect the mutation that's associated with this disease or trait, or did we not? That doesn't necessarily mean like we detected this disease, this dog will then go on to develop this disease. That is a different kind of jump that depends on more factors. It depends on the environment, it depends on the breed, it depends on the individual. So as far as our ability to tell you, will this dog actually go on to develop this disease? We do have some statistical measures to help us predict, but that's not going to be perfect. But for actually detecting the mutations, well over 99%. We have a bunch of redundancies in place. We have multiple probes that are all looking at the same mutation. If any of them don't agree, we don't make a call. We'll have a geneticist hand review it. For the ancestry, that is always going to be a bit more of a descriptive science than with the mutations that are directly associated with a disease or a trait. And that's because we did rely on people submitting samples of their purebred dogs in order to create that reference panel. And as we all unfortunately know, sometimes things get lost, sometimes papers get hung, things happen. So sometimes that's not perfectly perfect, but we do have the most robust reference panel in the industry, and the accuracy is reportedly about 98%. Ooh, can previously submitted samples be evaluated against newly discovered markers? And again, the answer is it depends. So if the dog was tested on a reference panel version that included that mutation, which sometimes the answer is yes, and sometimes the answer is no, then potentially it can be backfilled to include that new mutation. If we don't have that mutation on that chip version that the dog was tested on, we're not going to be able to return a result for it. So in that circumstance, we'd need a whole new cheek swab, unfortunately. But we do that at a discounted rate. So if you do ever need to do that, definitely reach out to us and we can do it for a little bit cheaper than doing a whole new test. Right, I think I'm through my whole list here that I got from you guys, which is awesome. Any other questions? I do have to go get my dog from the trainer in a few minutes here. So unfortunately, I don't too much more time, but happy to hang out and answer any other questions.
Well, I do want to thank you very much for your time. I know, just so you guys know, typically Dr. Dockweiler does not do these type of presentations. So I just want to thank her in advance for really meeting with us, because we are trying to figure this out. We've obviously tried to make this connection with Embark so that we can, obviously improve our breed and look at our breed. So I really want to thank you for that. And I want to be respectful of your time. I see there's one more question, but after this one, how about if we let you go after that? But again, thank you so much for your time.
Yeah, absolutely. I'm happy to be here. I mean, I grew up with pointers, so I have a soft spot for this type of dog. And obviously I have gun dogs now, but happy to answer these questions here. So if your pedigree and your Embark-based COI are both 22%, are they the same? And the answer is yes, to the best of our ability to tell. That genomic measure and that pedigree-based measure are going to be the same. I'm guessing that's a pretty big pedigree. I know that my dog's 30 generation pedigree is pretty similar to her Embark-based genomic COI. So the more generations you have, the better your pedigree-based COI looks like, is more accurate. And then would there be any benefit to submitting health certifications with DNA samples, for example, a thyroid test? And yes, absolutely. We do have a spot in your profile to upload documents. So that is really, really helpful for our scientists. And I know one of our scientists is looking at thyroid. So that one in particular would be a great one to have for sure.
Well, again, thank you
Welcome
so
to kind
much.
comments.
Thank you. Yes, this has been wonderful. Thank you so much for your time and sharing all this information with us. This is really helpful.
Absolutely. And please reach out anytime. I will get back to you on the number of dogs required for Matchmaker.
Okay.
Anything else that I can get back to you with? Any other questions?
I think I would just say if anybody has any questions that you didn't get a chance to ask or that comes up later, feel free to email us at contact@poodlepointersociety.org, and we can, forward those questions on to Dr. Dockweiler and keep in touch.
Yeah, absolutely.
And again,
Awesome.
thank you so much.
Yes, thank you.
Thank you guys for
Thank
having
you.
me.
Yeah, thank you.
Bye.
Daryl, thank you for getting this thing going. I think that was a very, very good thing for us.
I just appreciate her doing this for us, and thank you guys all for joining. We had 32 people on, and so thank you all. So
One of my big takeaways of this is that we really need to make sure we send in the phenotypic data to get some meaning out of this. Because, with 100, 200, 300 dogs, exactly what data besides disease data, of course, is the easiest one to do, but there's, you get some genetic test for a coat, and if the coat's really good, they need to know it. If the coat's crappy, they need to know it.
Yeah, for sure. For sure.
So
Big data is really, really helpful. And we appreciate
I think that's on us to get that phenotypic data to Embark.
Yeah, well, let's start working on it.
I think that's a great point. I think that's something we should encourage all of our members to do.
Yeah, definitely.
and I think, John, one of the things we probably have to do is really explain that to our members, that when they get the Embark test, talking to them what that actually means.
Yeah.
And how to send that in and how it'd be helpful. And then we can work with Embark on how to do that. So,
And it's a process over time, it's.
Mhm. Yep.
Yeah, Daryl Lee Branch here. That's a real key point here. I'm wondering how you actually do that. I mean, you get your breedings there, and you have an outstanding results, or you have a really poor results, and what would be the mechanism to convey that to Embark?
I think what we would try to do, and I know, Jenna, you got to get going, so I don't
Yeah.
want to keep you on. This might even be a conversation here. I really want to be respectful of that. I think we could ask that of Embark, how would they take that information so they could start collecting and helping us with that. But I think it's probably something we'd have to work with Embark to figure out. And while you're on, Jenna, if you don't mind maybe answering that.
Yeah. So I think the
Yeah.
surveys are probably the best place to upload it. Of course, they're paused for maintenance at the moment, but hoping to have it back in the breeder accounts in the next month or so. So that would be the best place for all of that data. And you can fill it out. It'll remind you to fill it out once a year, but you can fill it out as many times as you want. Like if something develops, you can certainly fill it out again. Your old responses aren't deleted. They're still saved so that we can still go back and look at them.
Great.
Mm-hmm.
Well, we'll definitely get that information out to our members and our breeders and try to round that database out.
And what we'll do here, guys, we'll let Dr. Dockweiler drop
Yeah.
off, and then what we can
We
do
know
is
you got
continue-
things to do.
Well, and we can continue the conversation as we want as a group to kind of,
Sure.
maybe talk about some of those things as a breed, club, where we're going and what this means. But again, thank you so much, Dr. Dockweiler.
Yeah, thank you all for having me. You guys have a good rest of your evening.
Thanks, you too.
Bye-bye.
Bye.
Any thoughts, group?
Yeah.
I was just
Thanks
so
you
know,
guys for participating too. This is great. We've got a great turnout. And thank you, Daryl. Really.
I have a question.
Yeah.
I was really excited to get her on here. Just,
Yeah.
she typically doesn't do these. I kind of maybe guilted her into it a little bit. That's why I want to be really respectful of her time.
Yeah.
But I think this is a good foundation for us to continue this conversation and how maybe we do these things, whether, we all look at breed mate and different pedigrees and how to match them. It is really appealing to look at that more advanced thing to compare studs would be really be an interesting piece to do to see how that helps the breeders overall when they're looking at selecting dogs. I think there's some really cool potential here.
Yeah, certainly. This was very educational and very helpful, and I think we can, use this information to build on, what we can do for the breed.
For those of us that may be less computerly inclined, could we put together like a cheat sheet on how to get to that partner thing, or the matchmaker and all that stuff? Is that free or do you have to pay for each one of those? How does that work? I haven't been able to. I haven't really looked too hard for it, but
If you're a member, you have access. If you've submitted Embark test that where you have your profile and the dogs that you've submitted and you own, you have access to all that. And Calvin, if you need me to, I can put you together a coloring book on how to do it.
I love it. I'm okay with the coloring book.
Yeah, we can certainly put together kind of a navigation chart for how to get around in the Embark stuff with screenshots, or we can just do like a video on how to navigate around in there as well. So yeah, that's a good suggestion, Calvin.
And no jab at Calvin. I will say that their website's not the most user friendly thing for someone that's not that's not techie.
Yeah. No, I struggle to get around in there too. And actually, I know some of y'all, like, I mean, I have all my dogs and they're related, and I test all my litters and stuff. But some of you guys have like, oh, this is this dog's mother, this is this dog's father. I don't even get that, and I still can't figure out how to get it. So I got to talk to somebody and try to.
Hey, just one point, guys, before we get too many other people dropping off. We still do have 18 tests available at a very discounted rate if y'all are interested. And it's on our website. It's on the member swag shop and all that stuff, and we can send a link out for that also. But just want to reiterate that point. We did purchase 50 more tests this year, and we have 18 left.
I think my biggest takeaway from this meeting was that the value of this information comes from the value of the data that they have, and the more that we can provide them, the more feedback we'll get.
Yeah, I agree.
Well, and it's been a couple years since I played around with which I think this was before they had the matchmaker feature. It was just their version of BreedMate, basically, their pair predictor. And if I remember correctly, which we don't have enough dogs in the database for that feature yet.
Yeah.
But once we do, because I attempted it with Adam Mayo, he shared me Deacon's DNA. And from what I remember, all you have to do is share access through email address to both parties, and then you have access to the pair predictor. But unless they've updated the website since then, prior to that, it doesn't give you the option for that feature.
Okay.
Yeah. And I know Jason Newberry has had Trench, and I've had Noonan done. I haven't shared, but I do know Jason knows how to do it. I've got to connect with Jason at some point to figure out how to share, because I know people have asked me to share Noonan's data. I'm not techie, kind of like Calvin. I might need a coloring book. Mine might need the numbers in there to know what colors go in there. But
so Daryl, if you go not under your profile, but if you go under Noonan's dog profile in your profile, it should give you an option somewhere on that page to share out. And I believe, which if you're doing it on a desktop, I believe all it asks for is an email address.
Okay.
And don't hold me to that. They could have updated this. This was 2.5, 3 years ago when I did this.
Okay. I'll take a look at it when I get home here tonight.
Daryl, essentially you have to add the person that you want to share it with as like a co-owner to the dog in the dog's profile.
Okay.
I like the idea of doing that group sign on. So maybe we'll try to figure out how to do that. That way we can all share, and then we can get access to that.
Yeah,
I like that.
really cool.
I like that. Yeah.
Yeah. I think that's a great idea for everybody.
Yeah, that would be a really good thing to do. Yeah.
All right. Well, I'll start looking into that.
All right. All right. Well, thank you all. I'm going to head out, but if you guys want to continue, by all means do. Thank you guys so much. I really appreciate you all joining in today. That means a lot.
Well, again, Daryl, thank you for
Thank
coming
you.
up with this and getting her. She did a fantastic job, I thought. Thanks for everybody that participated. Appreciate it. Appreciate every one of you.
Yep.
Thanks, guys. It was a great turnout. I hope you guys all appreciated it and got a lot of information out of it.
And we
if
have
you-
another one in a month.
Yeah,
Another
and if you
roundtable.
guys have any questions, follow-up questions that you want us to forward to Dr. Dockweiler, shoot us an email at the contact@pudelpointersociety.org email and let us know. All right.
All right. Bye.
Good
Have a
night.
great night, guys.
Good
Yep.
night,
you.
everybody.
Okay.
Bye.
Thanks, Sarah.
Hey.